Hello everyone, I am Ashik Mahmood. Thank you for joining with us to this ISCB-SC webinar. I would like to remind you, if you want to know about the upcoming webinar or how you can present your research and be part of this initiative, you can follow us on Twitter. Today we have an interesting talk by Mr. Simon Hebrew about an in silico approach for antimalarial therapy target, and he is a fresh graduate student of Kenya. If anyone have any question, just give a comment in a comment box. Now, please, Mr. Simon, start your talk.
Thank you, Mr. Mahmoud. As they said, my name is Simeon Hebrew. Recently finished my undergraduate course in biochemistry and molecular biology at Kenya. And just like to take this opportunity to share with you my study on the in silico profiling of a Plasmodium falciparum protein called CBP1 as a potential antimalarial therapy target. So in terms of antimalarial therapy target, we're talking about both as a possible drug target or as a possible vaccine target. So just to give you a background on where we are in terms of malaria, I think the most key thing is to interrogate the life cycle of the parasite. So as we know, Plasmodium falciparum, which causes severe falciparum malaria, has two hosts, the mosquito, which is the vector, and the human, which is the main host. So things to note, what happens primarily is when a microgamete and a macrogamete, which are two male and female gametocytes, bind together, they form a zygote, which propagate in the salivary guts of the mosquito to form a oocyst, which then differentiate from sporozoites that are then transferred to the human in form of sporozoites. So what happens is when a mosquito takes a blood meal, it takes in the sporozoites into the blood, and these sporozoites later move into the blood system, and majorly occupy the hepatocytes, which are the cells in the liver. So they also differentiate further to form merozoites. The merozoites themselves, once they enter the liver, they invade the red blood cells and they infect these red blood cells. So this is going to be our main target of the study: what happens in the infected erythrocytes, and how that can be antagonized in terms of making a possible therapeutic drug or vaccine. So, as I've said, my study was concerning the infected red blood cells at the merozoite form. So, what happens with this protein is that it is made by the mosquito parasite in form of an adhesing protein. So when the merozoites invade the infected red blood cells, they generate more copies of the protein, and they also make machinery that will aid the transport of the protein from the Paristophorus vacuole to the surface of the infected red blood cell. So these mechanisms or structures that are used to ferry the proteins are known as Morus cleft. So CBP1 is a very essential protein for Plasmodium falciparum because it aids the propagation or the cytoadherence of the infected red blood cell to the endothelial space on the blood vessels. So it is like putting a sellotape on a wall. It means you're really glued to it. Nothing can remove it until you use force. Now, in terms of malaria and its pathogenesis, when this protein attaches itself to the blood vessels, it causes blockage. And as we know, blockage of the microvasculature can later cause problems like hypertension. So I've talked about the protein. So what it binds to is what is found in the name of the protein. It's called CX3CL1, which is a chemokine. As you all know, chemokine are chemoattractants. They attract they are used to attract immunological machinery during an infection. So if it binds to the chemokine, it means it will diminish its activity as a chemoattractant because it's both in membrane bound and soluble form. So as I've stated, sequestration and rosetting are the scientific names given to these events. Sequestration being the fact that the infected red blood cell will now bind to the endothelial space and cause blockage as seen in the image, and rosetting meaning that the infected red blood cells themselves can bind to each other, and so that will eventually clog the microvasculature. Another event that is characterized by sequestration and rosetting is that the infected red blood cells cannot be cleared by the body. Remember that damaged red blood cells, usually cleared by the body using splenic clearance. So these infected red blood cells, once they bind to the CX3CL1 chemokine on the endothelial membrane, they can't be cleared, and hence they continue binding up in the body of the patient and later causing severe types of malaria, such as cerebral malaria and placental malaria. So this study was aiming to examine this protein using an in silico approach, in silico implying both bioinformatics and computational biology. We wanted to see whether it can be a viable drug design target or a viable vaccine target, and all this will be supported by the evidence that we collected during our study, because from what we know, it is an essential Plasmodium falciparum protein that is key for its pathogenesis. So if we are able to antagonize its activity, then we are able to suggest ways of actually indirectly curing severe falciparum malaria, which causes a lot of mortality, especially in sub-Saharan Africa, where I'm from. So key to note is we are working with 3 objectives. So we narrowed down our in silico approach to target 3 objectives. The first one of which is to determine the presence and roles of the conserved regions in the protein, because if we are able to learn about or discover the conserved regions, then we will be able to infer conserved functions that we can be able to antagonize at therapeutic level. The second one was to determine possible ligand-binding sites. So if we're able to determine whether the protein has small pockets for small molecules to bind to it, it means it is a viable drug target. And the third one was to assess whether it has genetic variations of polymorphisms. So we knew that the protein was a single copy gene. So we just wanted to validate whether it is subject to change over a period of time, or it is a rather stable, in quote unquote, protein which we can easily target for a vaccine or a drug. So in terms of my experimental approaches, just to give a quick overview of what I did, we were basically retrieving orthologs from a platform, a bioinformatics database known as Plasmodium. This is a database that is dedicated to genomic analysis and genomic data of all Plasmodium species. So it gives the gene ID, and you can be able to access the chromosome number, the exon, number of introns, length of the protein sequence, DNA sequence, and other details such as polymorphism, though at a less detailed level, because it is mainly used to retrieve sequences. So from Plasmodium, when you have the sequences in line with our objectives, we could either assess for conserved regions using a database or a software known as motif search. Third one will be to predict its structure, because after we predict its structure, then it means we can be able to detect the ligand binding sites. So we did this using ITSI and FIRE2, both of which are folding grading algorithm databases. And the third one of which polymorphism analysis was done using malaria gen, which I will explain further as we move on. So just to take you through the methodology, conserved region identification was done using Plasmodium and motif search, as I mentioned. So what we did is, by keying in the gene ID of the protein, we were able to retrieve orthologs from Plasmodium. We retrieved a huge number of orthologs, but for accuracy reasons, we were able to take down the number of orthologs we had based on length similarity of the sequences. And after that, in FASTA format, we fed them into motif search, and using a cutoff score of 1.0 E value, we were able to assess and identify the conserved regions, most of which were domains in this case. So for the ligand binding site prediction, as I mentioned, we only could have detected where the ligand binding sites were, where these pockets were, after having predicted the structure of the protein using an silico approach. So what we thought best to use was a fold threading algorithm using databases such as ITESA and FIRE. So I know there's a lot going on the screen, but just to summarize, fold threading algorithm works in this way. When you have a protein, say for example, our target protein is CBP1, what it does, it aligns the folds of the protein. We know proteins are made up of folds in the structure, so it aligns folds in the protein to templates present in the database to see whether there is any type of similarity, and then it comes up with a final model, the one which has the lowest E structure to use as your predicted model. So we had to do this using two databases for validation reasons. So we used ITESA first, and then we went on to use another database known as FIRE2. We generally preferred a fold threading algorithm to a homology-based modeling because I think for fold threading algorithm, it interrogates both the sequence and the structure of the protein for prediction reasons, and for homology modeling, it only interrogates the sequence. So we thought that fold threading algorithm would be more accurate to use in this case. And we were able even to infer transmembrane regions within the protein using another database known as TMHMM, which uses a hidden Markov model algorithm to detect transmembrane regions whilst showing only these regions and excluding signal peptides, which were not of interest in this case. So as I said, I will mention, talk a little bit about malaria gen. So I think this one is only matched on with people who work on malaria bioinformatics, as I did. So it is basically a community project database that was built to reach out to enable guys to network who are working on malaria bioinformatics. And networking in this case means they were able to insert or input data on nucleotide polymorphisms in Plasmodium so that they will be able to assess variation levels within different populations. So it is a really easy to use database. All you need to do is to just scan the gene ID, which we had. As you can see, it is PF3D70113900, gene ID containing the chromosomal location as well. So you just key in the gene ID and you're able to get allele frequency results. It's a global allele frequency, non-reference allele frequency, reference allele frequency. So it's a whole vast array of information that you'll get with malaria gene. So I will discuss my results as per each step that I've talked about. So for the first one, in terms of phylogenetic analysis of our orthologs, we generated this phylogenetic tree using Mega7, which utilizes a maximum likelihood method for phylogeny. So as you can see, for the orthologs, we are only able to get orthologs under species that are under the Liverania subgenus. This means that these were primarily primates. We have a human, for prayer, falciparum, and allele, we have a gorilla, and for Echinoe, and carboni, we have a chimpanzee. So of interest is that we were not able to get any rodent orthologs for this parasitic protein. And to us, that inferred that the fact that there are no rodent orthologs meant that the protein is actually very essential to pathogenesis in mammals. So maybe after evolution, after the evolutionary process, the protein was conserved in mammals. And then later on, because as you have noticed, we didn't find any human parasites as well. We only found Plasmodium falciparum in the ortholog list, meaning there's no Plasmodium vivax and the other human parasite, Plasmodium parasites as well. So this means that even after evolution, the parasitic protein was conserved to the mammalian species, that is the humans now, meaning that it plays a really crucial role in facilitating the pathogenesis of severe falciparum malaria. So this actually, it was just a proof of essentiality, and we also moved further to prove the essentiality of the protein by interrogating a database known as Phenoplasm, where we were able to see that it is impossible to actually silence or actually knock out the gene that codes for CBP1. So if you're unable to knock out the gene using a piggyback transposon model, it means the protein has a high likelihood of being an essential protein. So it means if we have an essential protein now, it further validates our claim that this protein is actually very key in the pathogenesis of severe falciparum malaria, and then moves to validate our objective as well, which is that the protein can be used to antagonize pathogenesis of falciparum malaria using different therapeutic agents. So if we to look at the genomic characteristics of our CBP1, I drew this schematic figure just to show the distribution of different regions. I think to note in this image is that there are actually three domains, conserved regions. Remember, we did conserved region analysis using motif search. So we discovered three domains. The first one is the ABC transporter domain, ABC standing for ATP binding consent transporter domain. So this domain, what it does, it majorly facilitates the intake of nutrients into the protein. So we're talking nutrients from macromolecules, such as polysaccharides and proteins, to smaller molecules, such as ions and carbohydrates. So I'm able to find out again the occludin homology domain, which is hypothesized to facilitate protein-protein interaction. So we might think that this is a protein that facilitates the binding of the CBP1 to the CX3CL1 chemokine, and the DNA binding domain as well, which is obviously to facilitate methods of binding DNA structures that encounter the protein. So of interest were the occludin homology domain and the ABC transporter domain, because if we are able to antagonize these domains, it means, first of all, we will create a deficiency situation for the protein, meaning it can't transfer in nutrients. So it's in quote unquote starving the protein. And also, if we learn how to possibly antagonize the occludin homology domain, it means that we are able to antagonize protein to protein interaction of the parasitic protein itself. As well, as you can see from region 182 to 190, which is shaded in green, is the extracellular region. We're able to get the exact array of sequences of amino acid residues which characterize this region. And as you can see, it is 8 residue extracellular region. And the red box is just to show you how conserved it is, meaning it is a very key region for which we are now hypothesizing is the main link between the infected red blood cell and the ligand. So it is the one that actually makes the infected red blood cell act as an adhesant to the ligand. I think to note is when I mentioned about conservation, someone may ask why we have a variation in the threonine and serine, but the threonine and serine are similar in terms of their physicochemical properties of them being both hydrophobic. So it will mean that the region has a really conserved role in allowing association of the infected erythrocytes to the ligand. So moving to what we got after protein structure prediction and the ligand-binding sites. So we were able to discover three ligand-binding sites. The first one of which was a leucine residue at position 186, a lysine residue at position 187, and an asparagine residue at position 188. So 3 residues, all located on the extracellular region, and all with good C-scores. So the C-score in this case means confidence score. As I mentioned, we used ITESA, and ITESA usually has its own set of statistical scores. So in this case, confidence score is a score that is given to show the quality of the predicted model. It just allows one to statistically infer how good the predicted model is. So it ranges from -5 to 2. And as you can see, our C-score was 0.03, which is statistically relevant and not badly off. We then moved to further assess the quality of the predicted model, because if you have a good predicted model, a protein structure prediction, it means you can easily move on to further high throughput protocols. But if you really don't have a good predicted model, you can fall away in drug design. So we used Ramachandran plot analysis, which Ramachandran plot just shows the stereochemical quality of the protein based on residue-to-residue geometry, and as well as the overall structural geometry. So it has different statistical parameters, but just to note for this presentation is that it usually has a threshold called most favored regions, which has to be more than 80% for it to be a good model. And for us, our non-glycine and non-fluorine residues in the most favored regions was 77%. So 3% of the 80% threshold, meaning it was a very good quality model. And we did this using software or informatics database known as ProCheck, which allowed us to infer that it was actually a pretty good model. 77% in this case implies that we had 177 residues out of the 243 residues, which were actually in the most favored regions. So just to talk about the pocket regions of the ligand-binding sites that we found, I think of interest for us was two features: whether these sites are conserved and whether they are sensitive to mutations. I'm going to start with whether they are conserved. As you can see, we were able to get a scale from FIRE showing color annotations from the lowest conserved regions to the highest conserved regions, that is from purple to red, if I'm not wrong. And as you can see, I've circled in blue the regions where we had the ligand binding sites. And if you compare that to the color scale, it actually shows orange, which means that the ligand binding sites are highly conserved. This means that during overevolution, they have not been subject to any selection pressure. So it means even over time, they can be a valuable target for small molecules to bind in terms of, now we're talking about pharmacology, research, and drug design. So it means that they are well conserved, and over time, they can be targeted. Secondly, for mutational sensitivity, what we have is another scale from blue to red, and the annotation, as I have circled in the ligand binding site, as you can see, what we had in terms of color was a green, yellow, and orange assortment. So it actually shows, according to the scale, that they are not sensitive to mutations over time, meaning that it is very there's a very low possibility of finding residue substitutions over time. That also supports the fact that these pockets are actually able to be targeted in terms of small molecule design and in terms of high throughput protocols that are going to be carried out in the next analysis. So we have highly conserved ligand binding sites and sites which are less sensitive to mutations. So that was a really good find for us. So just on the last analysis that we did, concerning the polymorphisms and genetic variation, as I stated, we used malaria gen, and after keying in the ID, we were able to get all this information. So to note is that we had 19 single nucleotide polymorphisms, abbreviated as SNPs, 11 of which were non-synonymous and 8 of which were synonymous. Obviously, of interest to us were the non-synonymous ones, and over the non-synonymous ones, we had a global allele frequency of 0.005, which is really low, meaning that these are not SNPs that are expected to cause any significant change in the protein structure or in the amino acid characteristic of our parasitic protein. So a very interesting find that we got is that there was a residue substitution that was occurring at the asparagine ligand-binding site. If you can remember, we discovered a ligand-binding site at the asparagine residue in position 188. So according to our find is that there was a possible asparagine to serine substitution at position 188, and we had to confirm whether this would alter our ligand-binding site. So we did in silico mutation. In silico mutation in this case involved using bioedit to physically change the residue in the sequence profile of the protein. So we replaced an asparagine with a serine, and we found out that after modeling the sequence again using fire and ITESA, we were not able to get the same ligand binding site at that position. So that means that in the case that this substitution occurs, the pocket actually disappears. And from what we are able to get is that possibly it could be caused by alteration of pH, which is needed for conformational change of the protein during folding and attachment to the ligand. But however, I just wanted to say that this substitution has a very low allele frequency of 0.005 and was discovered in Papua New Guinea. So it is not discovered in areas where falciparum malaria is severe or has a high endemic rate, say like Sub-Saharan Africa, Southeast Asia, Southwest Asia. So it means it is reserved to one region and has a very low allele frequency. So it is not something that we expect to trigger polymorphism or variations over time, such as we have seen with COVID, where we've been having variants arise from different regions of the world. Say we've had a variant from South Africa, from the UK, and from Brazil. So that will be actually statistically significant because you're having different variants which are composed of different substitutions at amino acid residue level. For example, for the UK involves a tyrosine residue replacing an asparagine residue at position 150, or the South African one is different, so that can be statistically significant. But for this case, you only had the substitution being reported in Papua New Guinea, so it does not raise a great alarm, but we have to count it in as being significant in our study. So just to answer the question at the end, so is our protein CBP1 a valid antimalarial drug and vaccine target? I think with the evidence that we're able to get, we can be able to infer that yes, it is, because we have an extracellular motif, which is maybe responsible for the protein chemokine interaction. So having an extracellular motif can help us even identify the ligand in the chemokine that is responsible for cytoadherence, and that will also aid therapeutic approaches and further research into the study. We're also able to discover conserved protein domains, as I mentioned, the 3 of them, and we talked about how antagonizing them can actually cause deficiency of nutrients to the protein and also interfere with protein-protein interaction of CBP1. We also discovered 3 ligand binding sites for small molecules to bind, which is a very key finding in terms of drug discovery. And lastly, as I mentioned, it has relatively low polymorphism. As I said, it is a single copy gene, so it is a better drug or vaccine target over time as compared to multiple copy genes, such as PF, EMP1, which is actually also an adenosine like CBP1, but is characterized by more variation as opposed to CBP1. So some recommendations after this study will be, now that we have validated that indeed this protein can be able to further our study in terms of now looking for a therapeutic agent to antagonize it. I think first of all will be to propose a high throughput screening or protocol to identify potential compounds that can provide a platform for drug discovery methods to begin. Also, since our study was in silico, it was bioinformatics and some bit of computational biology, it was dry lab. It can then proceed to wet lab so as to validate the in silico evidence. And also, since this parasite codes for a lot of proteins that actually work in this kind of, actually facilitate this kind of pathogenesis, it is key to establish a sequence database that has sequence profiles for all these malaria exported proteins, which are known as malaria exportome as shown, so that we can be able to carry out different analysis of them, see whether these are potential vaccine or drug targets over time, and now move to antagonize the targets as well. As we all know, for malaria right now, we have a vaccine, which is called RTS, which is only 40% efficient. So over time, and due to causes of drug pressure over time as well, it is necessary for us to come up with more targets for drug and vaccine approach, which are more efficient, and as well, even in the rise of antimalarial drug resistance, we need to come up with more targets that are less polymorphic and can be valid over time. So I want to thank my supervisor, Dr. Daniel Kiboi from Jomo Kenyatta University of Agriculture and Technology in Kenya, and Mr. David Barra from the Pan-African University from helping me carry out the study. I think they were really instrumental in the design and also giving comments in the study, and as well for the International Society for Computational Biology Student Council for organizing this webinar and giving me the opportunity to present my work. Thank you.
Thank you, Mr. Simon, for your amazing talk. So, any question from audience? Write in comment box.
Maybe you have a question.
I think there is no question. No one. I have one comment here. She is Kayoma. He says that my brother.
Yeah, thank you. He's my classmate.
Okay, nice.
Do you have a question yourself?
No, everything was really clear. It was really understandable, and I think no one have a question. Then thank you, Mr. Simon, for presenting the webinar. For a small reminder to all, we arrange 2 webinars per week. To stay connected with us, just follow us in Twitter. Good day and stay safe.